Hi, and welcome to the part 2 of the study of prokaryotes. In part 1, we had looked at the characteristics of prokaryotes, the taxa, a few examples, and the external structures of the prokaryotes. So now we will look at the cytoplasmic membrane, which is the interface between the outer and the inner environment of the cell, and then the cytoplasm, and finally the internal structures of the cell. We will briefly look at the schematic overview of a prokaryotic cell. For that purpose, we will focus on the structure of a bacteria. In this schematic, we can notice the plasmamembrane in this dark blue line surrounding the cytoplasm, the cytoplasm in this light blue color, the internal structures such as the ribosomes shown here in red, and the inclusion bodies shown in purple. As we have studied before, the nucleoid is the region where the DNA is located, and the plasmid is an extra chromosomal DNA present in some bacteria. Now we will focus on the cytoplasmic membrane. We will look at its composition and function and at the differences between bacterial and archaeal cell membranes. Bacterial cytoplasmic membranes are made up of phospholipids and proteins. It has a phospholipid bilayer structure in which the phosphate heads shown here in yellow are hydrophilic or water loving, and the hydrocarbon tails are hydrophobic or water fearing, facing each other on each side of the bilayer membrane, and here they are shown in green. The proteins found in the membrane can be integral protein spanning all through the full layer of the bilayer membrane, or they can be on each side of the membrane, known as peripheral protein. The main aspect of the cell membrane to note is that it has a fluid mosaic model. This means that the membrane is dynamic with the movement of proteins from one place to another within the membrane, and with the flip-flop movement of the proteins as well. The function of the bacterial cell membrane is similar to that of all biological membranes. They include selective permeability, concentration gradient, electrical or electrochemical gradients. Also, the cell membranes aid in the transport of solutes and solvents through passive or active transport. Shown here are examples of a hydrogen ion gradient in which there is a higher concentration of hydrogen ions on the outside of the cell compared to the inside of the cell. This concentration gradient is maintained by an ATPase enzyme that is present on the membrane. The next example is that of diffusion. In diffusion, solutes such as ions diffuse from a higher to a lower concentration gradients. Examples are the chloride ions shown here in green, and the sodium ions shown in blue moving from the outside of the cell to the inside of the cell, and potassium ions shown in purple moving from the inside of the cell to the outside of the cell. What is to be noticed also is that there is a net positive charge on the outside of the cell over here and a net negative charge on the inside of the cell. This is known as an electrochemical gradient. In osmosis, water moves from an area of less solutes, of less solutes to an area of more solutes, trying to equilibrate the water concentration on each side. Finally, one special function of the cytoplasmic membrane in prokaryotes is that of energy metabolism. We had looked at differences in the bacterial and archaeal cell walls. Now we will look at differences between bacterial and archaeal cell membranes. While bacteria and actually eukarya use fatty acids as their side chains in the phospholipids, archaea use the phytanyl side chains, which are branched, as you can see here by these projections. While bacteria and archaea as well have a bilayer, as shown over here, 2 layers on each side, archaea have a monolayer cell membrane, shown as 1 layer. Also, the linkages that bacteria and archaea use are the ester linkages. Whereas the archaea use the ether linkages, shown here as CO, whereas for ester it is COO. Summarized here are the same observations. Archaeal cell membranes have branched phytanyl side chains instead of fatty acids. The phytanyl chains are connected to the glycerol by ether linkages instead of ester linkages. The lipid tails of the archaea are longer and form a monolayer instead of the bilayer. This has particular advantage for the archaea that are hyperthermophiles, since it prevents easy peeling of the membrane, which could easily happen for the typical bilayer of the bacteria. In this part, we will focus on the cytoplasm, which is composed of the cytosol and the bacterial subcompartments, also known as the internal structures. The cytoplasm is the gelatinous material found inside the cell and enclosed by the cell membrane. It consists of the cytosol and the bacterial subcompartments. So, if you remove all the subcompartments within the cell, all that remains is the cytosol. The cytosol is the semi-fluid mass of proteins, amino acids, sugars, nucleotides, salts, vitamins, and ions, all of these dissolved in water. The subcompartments are mainly inclusions, endospores, and non-membranous organelles. Inclusions are reserved deposits of nutrients or compounds. So, these are structures that store nutrients such as lipids and starch, and compounds such as nitrogen, phosphate, and sulfur. These nutrients and compounds are taken up in abundance when needed and used when they are scarce in the environment. Endospores are tough, dormant, non-reproductive structures used as a defensive strategy against hostile or unfavorable conditions, as when there is a drought or in extreme heat. They are also capable of causing disease. Examples of bacteria that have endospores are Bacillus and Clostridium. Finally, non-membranous organelles include ribosomes and cytoskeleton. Since bacterial compartments are the internal structures of a bacteria, let us study those in detail. We'll start with endospores and inclusions and end with ribosomes and cytoskeleton, emphasizing structure and function. Figures A and B in the top panel refer to endospores. In the top panel, figure A is a staining of endospores, as you can see them over here in these circles. Figure B is a schematic of the process of formation of endospores. It starts with a vegetative cell that divides unequally. The smaller part develops a spore coat, which then thickens until it finally becomes a free endospore. Endospores are resistant to heat, desiccation, and radiation. However, they can be destroyed by burning or autoclaving. Figures C and D in the bottom panel relate to inclusion bodies. Figure C on the left, you see a differential interference contrast image, or a DIC image. And on the left you see a fluorescence microscopy image. The dark spots in the DIC and the blue highlighted spots in the fluorescence microscopy image, and the green spots are all inclusion bodies within the prokaryote. Figure D is showing a staining of bacteria. The inset image is showing the outline in yellow of two bacteria within which are metachromatic granules stained in dark blue. Metachromatic granules are also examples of inclusion bodies. Bacterial cells have thousands of ribosomes. The size of a ribosome is expressed in Svedbergs units, and it is determined by the sedimentation rate. In prokaryotes, ribosomes are 70S. All ribosomes are made up of two subunits. In prokaryotes, the subunits are a smaller 30S subunit and a larger 50S subunit. The subunits are made up of polypeptides and ribosomic RNA. Because the sedimentation rate depends not only on the size and mass but also on the shape, the addition of a 30S subunit with a 50S subunit does not add up directly to 80S but to a 70S ribosome, as shown over here. The cytoskeleton of prokaryotes is the collective name for all structural filaments. It is the internal network of fibers. The cytoskeleton provides shape and movement. Now, finally, a summary of prokaryotes, part 2, and prokaryotes in general. In this part 2 of the study of prokaryotes, we looked at the prokaryotic cell membrane, which is a phospholipid bilayer with a fluid mosaic model. We saw the three main differences between the bacterial and archaeal cell membranes. We studied that the cytoplasm is composed of the cytosol and subcompartments, also known as internal structures. The internal structures include the endospores, inclusion bodies, ribosomes, and cytoskeleton. As a recap, by definition, the word pro means before and karyote means nucleus. Prokaryotes are those organisms that existed before there was a nucleus. Therefore, these microorganisms do not have a nuclear membrane surrounding their genetic material. Instead, the DNA is found in a region which is known as a nucleoid. Prokaryotes also have no membrane-bound organelles in their cytoplasm. Or, in other words, they lack a number of internal structures that are bound with a phospholipid membrane. The chromosome of prokaryotes is small and circular, double-stranded DNA. Some prokaryotes also have plasmids that are separate from the chromosome and replicate independently from the chromosome. Overall, prokaryotes are small and simple organisms when compared to eukaryotes. This concludes the study of prokaryotes.